Okay, so you start off with DNA, double-stranded, and then the enzyme helicase comes and it causes the DNA strands to separate. Then you're gonna have RNA nucleotides that are just gonna be floating around, and these RNA nucleotides will attach to one of your DNA strands. Okay, but remember that if this is DNA A, T, T, G, it should match up through complementary base pairing. But remember, the difference between DNA and RNA is that, in terms of bases, is that the A is replaced with U. So what you should have then is, the T is replaced with U. So it should be A with U, T with A, T with A, and G with C. Okay, so the green, what I'm doing here is RNA, and the red is DNA. Okay, and then lastly, what happens, so I'm going to do this again, U, A, G, and A, T, T, G, is you need RNA polymerase to come in now. So another enzyme, RNA polymerase, and this will create the backbone for the nucleotides. So can you see in this top one, the nucleotides are not connected, they're only connected to the DNA strand, but now I'm creating the phosphodiester bond, or the backbone. And RNA polymerase does that. So right now you've got one strand of RNA and one strand of DNA, but this is only there for a temporary time. What will happen is this DNA strand will then go away. So you're just left now with a strand of mRNA. So we've made this mRNA, yeah? But that mRNA that I showed you in the last video at the end is called pre-mRNA. And the reason why it's called pre-mRNA is because this strand is basically coming from this. But within this, you're gonna have coding DNA, coding parts of DNA, which is important in making proteins, but you're also gonna have non-coding DNA, which is not really important, okay? And what we call the coding DNA is exons, and the non-coding is called introns. So you only want to make proteins from the exons. So that means then that this pre-mRNA will also have exons and introns. So if I'm going to draw this, now let's say I draw this pre-mRNA as a box now, but I split the boxes up, or I split this box up into smaller sections, and this could be an exon, this could be an intron, this could be an exon, this could be an intron. What we then do, or what your body then does, is it wants to get rid of the introns and just leave the exons, because it will just make the process quicker. Because if you think about it, you want to make proteins very quickly, because you may need it for something like white blood cells, or like clotting your blood, or so many other important things. You want to do it quickly. So it goes through a process of splicing. And all splicing does is removes the exon, the introns, and keeps the exons. So that means at the end, if I cut this out through the splicing, and I cut this out through splicing, I'm just going to be left with two exons from here, yeah? But of course it's much longer than that. And this is what you call mature mRNA. So this now can then go to the ribosome and then go through translation, last part of protein synthesis. The first step, and this is what you can just write in your, when you're answering a question and you'll get the mark. You write mRNA attaches to ribosome. That's all you need to say for the first step, okay? So if I'm drawing a ribosome, the first thing that's going to happen is the mRNA will attach to it. And what it means by attach, it would just kind of enter the ribosome like that, okay? Then the next thing about the mRNA is it's going to have the bases. But if you remember, what we said is that 3 bases code for 1 amino acid. So we have to think of the bases in triplets now. And what do we call the triplets? We call it codons. So that's one codon, that's another codon. So you need to remember, and this is what you say for the second thing. There's a codon on the mRNA, or the mRNA is a codon, or the mRNA is split into codons. And why is it important now when we're talking about translation? Because what does the codon attach to? The codon attaches to an anticodon. And where's the anticodon coming from? It's coming from tRNA. So I'm just going to draw random thing here, and this is supposed to represent tRNA. tRNA. And the tRNA has 3 exposed bases, and this is what we call the anticodon. So if step 1 is mRNA attaches to ribosome, step 2 is codon on mRNA joins to anticodon on tRNA. So I would then do the tRNA here, and it's attaching to a codon from here, yeah? So that's step 2. Then, what does the tRNA have attached to it? The tRNA has a certain or specific amino acid attached to it. So this tRNA will bring this amino acid, so the square, and then on the next codon, let's say the next codon is different. So if I do here now A, U, G, and this is G, Z, A, there's our next 3 bases, or our next codon. So another amino acid is going to come along. Sorry, another tRNA is going to come along, and that should bring another amino acid to join there. Okay, so this one we can say is circle. And then that will just keep on happening. So that will just keep on happening on the along the ribosome, okay? And remember now that this is an amino acid, this is amino acid. What will then happen is these amino acids will join together, and that bond is peptide bond. And this is joined through condensation reaction.